So our project is all about Haloarchaea and the carotenoid expression. We call this the Pink Wacko Project, and this is by Sierra and myself. So you might be wondering, first off, what is the Pink Wacko Project, outside of the best name you've ever heard? So it all started when we were contacted by this artist who wanted to make this giant art installation. So it was an acre large pond that wanted it to be as pink as the Great Salt Lake. And while this is like a fun little story, it actually raised a very important scientific question. How do we actually enrich pink coloration?
So we kind of went all the way back to the beginning. So we started out with, why is the Great Salt Lake pink? And as you can see from this wonderful photo by our friend NASA, or if you've ever flown above the Great Salt Lake and looked down, I know not many people actively want to look at the Great Salt Lake when they're flying, but that's all right, that's all right. There are two different colors. There are the north arm, which is this bright pink color, and the south arm, which is this very green color. And as you can see from this photo down here, that very green color is due to microbial mats. And up here, this pink is due to the Haloarchaea that are found inside of the Great Salt Lake. Now, the Great Salt Lake is about 10 times saltier than the ocean, which is kind of weird to me, but very cool. And archaea are salt-loving, so they grow there and they thrive there. And what we found was that Haloarchaea have carotenoids. And these carotenoids provide pigmentation to not only the Haloarchaea, but also to things like carrots and fall leaves and tomatoes.
So how do we actually amplify this pigmentation? So we went and we dug up a lot of research, and it really focused on halobacteria. And not only that, but monocultures, going and trying to amplify the pigmentation of a single species, while we're more interested in a community, a polyculture, if you will. So we're kind of stepping out a little bit on our own here. So we wanted to start off on the right foot and to have a base. So we collected our own samples to test the differences in chemical composition to see if ions and nutrients correlate with high carotenoid composition. So we started and got our first sample courtesy of the Natural History Museum, who have a Winogrosky column, which allows you to visually compare the differences between North and South Arm water. And the pigmentation of the Haloarchaea really sends home this point. So we wanted a very pigmented sample.
And then we kind of were wandering around the tar ponds, and we found this very bubblegum pink sample, which we were like, this would be a great way to contrast our Natural History Museum sample. So we took a sample of that, and then about 4 weeks later, we went back to that same tar pond that was kind of that weird color, and we found out that it was darker and closer to our Natural History Museum samples. So as you can see in this bottom corner, that sample is very bright pink. That's the one that we took from our first sample, and then our second sample came from this this pigmentation where it's kind of a darker pink. And we found that quite interesting. So we decided to compare all three of those samples to see what was in them.
And we compared them in iron, phosphate, magnesium, and nitrogen composition, as well as their pH and percent salinity, with the exception of the second tar pond, but that's a different story.
Story for another time. So what kind of started this chemical compound question was the tar pits smelled awful. They smelled completely different than our Natural History Museum, and they smelled like sulfur, which sadly we didn't have anything to test sulfur in it with, but we were able to test a couple of different compounds. So as you can see from this graph, the blue represents the Natural History Museum, the red represents the first tar pit sample, and the yellow represents the second tar pit sample. The iron compounds that we tested. As you can see, the Natural History Museum was definitely higher than both the tar pit samples, and the second tar pit sample was higher than the first, which this indicates that there might be a correlation to pigmentation through iron, which also goes along with some of the background research that we did. Of course, that was all with Haloarchaea. And then there was this phosphate that we tested, and it doesn't appear to change pigmentation. As you can see, there is basically no movement from the tar pits, even though there was a change in color. There was no variation in the phosphate changes. And so because of that, there may indicate no correlation. And then the magnesium, we saw a decrease in it, which that one we definitely are kind of in the air with. We haven't quite figured that out, but there could be a correlation with it, but there's most likely not, just because the Natural History Museum was definitely higher than the tar pit, and the tar pit decreased over time. And then we had the nitrate sample, which that definitely indicates there's no pigmentation change due to the nitrate, because there was no nitrate in the tar pit samples. So the estimation for that is that it came from, most likely, the Natural History Museum, whatever they put into their column.
We also did the pH and the salinity as previously mentioned. So they had roughly the same pHs, but different salinities. The Natural History Museum being more salty than the tar pond, at least in the first time we took a sample from it.
So our next steps, because we definitely know that these chemical compounds may not be everything to determining what exactly causes this change in color. So our next steps are to kind of look at the species. And so right now we're running PCR to identify the different types of species, to find out what their best homeostasis state is, and what better, to look at than homeostasis in relation to temperature and these stressors in their environments. And so through determining what species we have, we can determine those correlations.
Through 96 well plates, we also plan to look at concentration and other compounds and chemicals. So concentration-wise, we'll be able to actually see the cell concentrations to see if it's just the number of Haloarchaea present that's actually contributing to the color, or if it's just they have more pigmentation. And that's where we can get the level of carotenoids. With the other compounds and chemicals, we want to test our compounds independently to see what actually causes a change, because from what we've done, we don't know if it is just one thing or a conjunction of multiple compounds that lead to different levels of pigmentation.
And finally, one of the things that we are doing, bringing it back to that art project idea, is we're trying to use this to not only further research in Haloarchaea, but we're also using this to further education through research. And so we've started creating links to that through doing the Pink Wacko projects with this lady, and our other project with the Winogradsky columns. We're making our own. And we're just looking at this to see how over time the water and soil change as the microbes segment to their happy place. So anaerobic microbes will settle closer to the sediment, and aerobic microbes, as you can see with this nice, like very pink line in there, they'll settle closer to their happy place, which is kind of further up in the water. And one of the things that we can learn from Winogradsky columns is that when it's pink, it doesn't always mean that it's archaea or Haloarchaea. In this case of this column, it definitely is, but we also took a few other columns, such as the Antelope Island, the Spiral Jetty, and the Farmington Bay, And in previous studies with Winogradsky columns, there have been purple algae or bacteria growing in Winogradsky columns from the Antelope Island samples. And so it will be interesting to watch those grow and see how they change. So thank you for your time. And if you have any questions, please feel free to ask. And also thank you to Bonnie, our lovely mentor, who has provided us with this photo and with so much help.
Oh boy, I'm not sure I should keep that up.
You have no choice.
I look so querulous. Okay, fantastic. Thank you. I'd like to open the floor for questions. Go, Preston.
So which part of the cell actually carries the pigment? Is it a structural component of the cell? Is it something inside the cell? And you know, why do these particular organisms have it and others don't?
So kind of what we know, I don't know if there's exactly a particular place, but I do know that there are carotenoid biosynthetic pathways, and those pathways give that color because they stop on specific and influence with specific genes. So these carotenoids are lipids, and they're not particularly the product of the gene, but they stop on different pathways and kind of give that influence of the color.
Okay, so if they're lipids, then are they part of the cell membrane? Is that where they reside, the pigments?
Yes.
Okay, thanks.
Of course.
They do. Another question, please. I have a question, like going off of Preston's, just I have, sometimes this hits me a bit, but because I'm not a real biologist, I don't know the answer. But it seems to me that organisms that live with a relationship to light are more pigmented. And so I've always been interested in the pigments because they may have a connection to the photobiology of these organisms. I mean, does that ring true to you guys? I mean, they live in the sun, right?
I just kind of think of like one paper that I read where when it wasn't exposed, when the cells weren't exposed to UV radiation, they actually had a higher rate of not having any pigmentation at all. So, like, I feel the connection.
Yeah, so there is a correlation between the amount of pigment they make and the light. So are you guys growing any cultures in the light yet? Oh, I guess you are with your columns.
Yeah, with the columns we do plan on adding more UV and growing them under UV in different intervals in the future, but that'll be a little bit down the road once we figure out the different types of Haloarchaea that we have.
Yeah, not to interrupt, but that was sort of the behind my question. I was wondering if the red pigment is in the cell membrane to protect the interior of the cell from high UV concentrations, if it's evolved that way. I have no idea, I'm just guessing.
On that, I haven't read any research on that. I don't know if Anna has, but I'm not sure. That's something we definitely would have to look into.
Yeah, I feel like it's yes, but I can't cite anything off of the top of my head.
Do you remember, maybe you haven't read Dan's paper. So I had a student who worked on UV and carotenoids, and he did find that to be the case, but for different reasons than we knew. It had to do with soaking up free oxygen radicals, which I never thought about in damage. Anyway, we can talk about that more from my point of view, but I don't want to take over your space.